Thanks for joining us today for the Tools and Technology Seminar Series. For those of you who are online, if you have questions, you can put them into the chat box and I'll be monitoring that and can let our speaker know. If you'd rather ask a question verbally, you could use the Zoom reactions at the bottom right and raise your hand, and then we can bring that up to the speaker too, so they know that there's a question being asked. So with that, I'm actually going to turn this over to Mike Cianfrocco, who's going to introduce our speaker today.
Okay, thank you. Hi, everybody out there on Zoom. So my name is Mike Cianfrocco, a faculty in the Department of Biological Chemistry and the Life Science Institute. And so we're, not in the sort of typical tools and development lab, I would say, but we're a lab that uses cryo-EM to determine protein structures. But the reason that we're here today is that I wanted us to share Eli Lee's work. So Eli Lee is a postdoc at my lab who does computational methods development. And we have this really cool new story related to cryo-EM data collection that I want to share with the department and the community here in general to get feedback. We want to advertise for potential grad students or postdocs or collaborators to think about how we can keep bringing all these very nice algorithms into cryo-EM in lots of different areas. And so with that, I'll turn it over to Eli.
So hello, everyone. So I'm going to share a story about accelerating structural biology is basically machine learning approaches to automate single particle cryo-EM data acquisition. We combine, so the framework is a combination of CNN and our reinforcement learning to help the microscope to steer to the good areas to automate the process and also to achieve higher efficiency. So it's an overview of cryo-EM data collection. Cryo-EM is a technique that has been growing very fast in recent years. And it's been invading the space of absolute crystallography. If you know anything about structural biology, it's just to solve the atomic or structures for large biomolecules. And the idea is cryo-EM at a high level is very simple. You just purify your sample and put it on this grid, freeze on this grid, and you look at it through an electron microscope. But when you zoom in, so this grid is about 1 millimeter in diameter. It's very small, but compared to single particle proteins or samples, it's very huge. So when you zoom in, there are a lot of squares because on top of the grid, and then inside each square there's a lot of holes inside each square. And we collect micrographs, what we call micrographs, which is the images we use to analyze later inside each of the holes. And from there you can see single particles of proteins or samples that you're interested in it. And then you analyze these micrographs and get a structure. So, so you start from a grid, your sample is on the grid, then you have a lattice of squares, and then you have a lattice of holes inside each square, and you collect one or more micrographs inside each hole. And then this is a simplified pipeline, and then you pick some particles from your micrograph, and you have a few thousand micrographs per data set you end up with a number of particles. You pin your particles through this 2D classification. You get 2D, basically get 2D class averages that you think has higher resolution information in it, and then use these particles from the pin to the averages to do the 3D reconstruction. So this is how you get a 3D volume or a 3D structure from the data on the grid. So a number of things could go wrong on the data collection side. And for the downstream processing to be easier, you want your upstream to be as clean as possible. So showing here is the good example of for the ideal case. You'll have a monolayer of your particles and your ice thickness is good enough. So you can see your particles and there's no aggregation, not too dense. But in many cases, it's either the ice thickness is ice is too thin, so your sample get denatured, or it's too thick, and the particle is too concentrated. You see all these aggregations, or not with high resolution information. or too dilute, basically you don't see any signals from your micrograph. And processing challenging data set requires clean micrographs from the upstream. So example is this GPCR data set. When we just do an initial naive analysis, pick the particles from all the micrographs. And because the particle picking process is not 100% accurate, you will end up with a lot of false positives. So after you do 2D classification, the 2D class average is like very noisy. But after we manually curate and discard 40% of the micrograph with visual inspection, we can end up with a much cleaner 2D class averages. So the better data quality in the upstream, the easier for downstream processing. And how is our data quality now? So from all the micrographs we collected from January 2019 to May last year in our lab, about 50% does not contain any high resolution information at all. So even though we try to collect as much as good data as possible, there's still a lot of data that's basically just doesn't contain any high resolution. And the efficiency of data collection greatly depends on expertise. If you're a new user, you'll end up with making a lot of mistakes during the data process, during the data collection process, and you waste a lot of time. And the question is, how do we collect as many as good micrographs in a limited time frame, and preferably without an expert steering the microscope, so anyone can just start the data collection, and there will be an algorithm to collect as many as good micrographs. And I didn't mention here, but some of you might know that cryo-EM is a very expensive technique. So the scope itself costs a few $1 million, and installing costs another few millions. And then to keep it running, if you combine all the costs, the cost per day is around a few thousand per day. So the efficiency and automation is needed in this field. But so cryo-EM grids are complicated data landscapes. As you can see from this whole atlas, image, the squares and holes has a lot of variability. So on this atlas, you see some are there are thicker eyes, and some are there like thinner eyes, or maybe just empty, or maybe the squares are just ruptured. And it really depends on the user to decide how to navigate through the grid to find the best way through. And also keep in mind that the grid is very big compared to the to your sample. So for a typical data set with a few thousand micrographs, usually you only end up image only 1 or less than 1% of the total grid. And you need to start from this atlas, and with other medium or lower magnified images, try to collect a few thousand micrographs that covered only 1% or less of the total grid. And you want those micrographs to be as high quality as possible. So so we're thinking to break this into two parts. The first part is to get a landscape of the data quality across the atlas, or at least across a part of the atlas. So here we're showing different layers of the different magnifications across this atlas. So this is on the right, this is the atlas image. And when you zoom in, you have like multiple squares. You zoom in one square, you can see all the holes in it, and then you take patch level images. And this patch has some ruptured holes. So we don't think these holes are good ones, and we think probably these holes are good ones. So we sort of predict the quality of the data on the whole level images, where we have more confidence. And we take all this information from all these different patches and overlay. We basically plotted them on the square level image and plot it back and plot it back. So ideally, what we want is to end up with an atlas with the data quality landscape plotting on top of it. So so the goal here is to collect as many as good holes in a limited amount of time. And we want to, the ideal solution would be we survey and assess a list of thousands of targets. So maybe a list of 1,000 targets is not enough to cover the whole atlas, but since we're only collecting low and medium magnified images, we save a lot of time by just assessing the quality of the whole level images. And we also want to balance the trade-offs in the time for switching to different patches, squares, and regions of the atlas with data quality. So on one hand, we want to optimize the data quality, collect as many as good holes as possible. On the other hand, so basically moving the microscope to one place to another place that are far away across the grid takes a lot of time. So we also need to balance the time of moving the microscope in this scenario. And also we want this framework to adapt to different samples and different grid types. So what we propose is reinforcement learning guided cryo-EM data collection. The first step will be we do a survey across the grid, and we assess the whole qualities and plug them back to the atlas image. And then we start to trajectory planning based on reinforcement learning. And from the trajectory, we collect exposures. And once you get that exposure, you know the ground truth of your data quality. Then if you are doing something right, then you get a reward. And then you learn your policy of trajectory planning. So this is the training process where you learn the data collection, the optimal data collection approach on this grid, which is also called the learned data collection policy. So there are several key questions. First is how do we define good or bad exposures? And we have an answer to that. And how do we assess whole quality? And we also we have a deep regressor, which is a CNN-based model to predict the quality of a whole from the whole level images. And 3 is how do we plan a path across the grid? And we use DQ network, which is one of the reinforcement learning methods that we use here. So first question, how do we define good or bad exposures? We use a CTF max resolution. And for those of you who are not cryo-EM experts, this is basically quantifying the signal at the Fourier space at which, so if you have high resolution, then you'll see more signals in the Fourier space in the high frequency areas. If your micrograph has lower, doesn't contain any like a high resolution information in the Fourier space, you don't see as many of the tone rings in the higher frequency areas. So the good thing about it is this is an unbiased approach, and generally it's correlated to data quality and very quick to calculate once you have the micrograph. The bad thing is that it's unrelated to particle quality and is also eye thickness dependent. So if you have, say, a particle with aggregation, in many cases you will get very good CTF resolution, but not a usable micrograph. But we just go with this CTF resolution because we think this is basically the best we can do to define or evaluate the good or a good micrograph. Next question is how do we assess whole quality? And the answer is we use a CNN-based, ResNet-based deep regressor to do that. So we take all the whole images combined with a ground truth CTF resolution and train a deep regressor based on that. So in the following experiments, we use 2 different regressors. First one is a grid-specific regressor, which is trained on over 13,000 of holes, and basically it's trained on a subset of the data set and tested on another subset of the data set. The other is a general regressor, so we also want to test the generality of this framework. So we train the general deep regressor that is trained on over 100,000 of holes and CTF max resolution pairs. And this is showing here, two different plots are the results for the regressor. So as you can see, ideally, you should all the points should be scattered along this diagonal line, but we don't. So because we are trying to predict very high resolution information from the pixel size with over 100 angstrom, over 100 angstrom. So it's anticipated that we don't get a very perfect result from our deep regressor. But I'll show later that you don't need to have a very good prediction from this regressor to actually do meaningful or actually collect efficiently for this framework. So the last question is, how do we plan a path across the grid and we use DQ network to achieve this? So for those of you who might know something about reinforcement learning, the reward table is what is optimized. So total reward is what is optimized during a time period, and the design of the reward table is very important. So we want to balance the quality of the data and also the time wasted or the time cost by moving when you move the microscope to a different area. So we defined that if you collect a micrograph in the same patch, it will cost 1 minute, and the reward, if the CTF resolution is lower than the threshold number, is going to be 1. So basically, if you're collecting a good micrograph according to the threshold number, then you get a maximum reward. If you don't, then you get a reward of 0. So similarly, if you collect a micrograph in the same square but not in the same patch, then we define the time used will be 2 minutes, and then you get a lower reward if you collect a good micrograph because you wasted some time in the movement of the microscope. And similarly, if you move to a farther away area, the time cost will be higher and the reward will be lower if you collect a good micrograph. For all the movements, if you collect a bad micrograph, which means the CTF resolution is higher than the threshold number, then you'll get 0 reward. So so the connection between the deep regressor and the polysampler is shown here. Deep regressor will output the quality score, which here is CTF resolution we use per square, per hole. And this quality score per hole will be combined with the hierarchical grid structure information. It's basically where this hole comes from. This hole comes from this patch, this square, and this grid. We take that all information and also its quality, and we engineer a multi-dimensional features and use that as the input for the policy network. So this deep Q network will output a Q score, and based on the Q score, it'll determine an action, which is where the algorithm thinks it should collect from the data from. And this action will change the states, basically will change the input of the Q network, and then it'll predict another action. And once you output an action, basically you want to collect data from this hole. And once you have that, you will also obtain the ground truth of the quality of that hole. So it'll also tie up with the reward. So the goal of the DQ network during training is that it'll maximize the reward in a given time frame. And what will output in the end is that the DQ network will output a series of actions. Basically, you have maybe a few thousand of candidate holes, and the algorithm will tell you like you should click on this hole first, and the next one, and then the next one. So it will output the trajectory of the hole that it thinks you should collect. So to combine everything together, cryo-RL first use this distributed data collections, where we want to have a survey across the grid, or at least several squares on this atlas. We collect patches and we identify the holes, and we segment all the hole images. And then based on different hole images, we run the deep regressor to output a quality score per hole. And also based on the hierarchical grid structure, we combine this information with a quality score and engineer a feature used as input for the policy network. And the policy network will output the action will basically output a trajectory of actions, basically a trajectory of holes that it thinks you should collect the micrograph from. So because this framework is not integrated in any real software yet, for testing cryo-RL, we need to obtain the ground truth to actually evaluate and to test the performance. And to do that, we need to collect a systematic data set. Basically, for example, in this data set of elderlays, we selected 25 squares and collected all the micrographs possible from the square, from all the holes in these 25 squares. So we end up with over 3,500 holes, and for each of the holes, we have a ground truth CPF resolution attached to each of them. And these are the example micrographs. Because we are not trying to incorporate any human knowledge in this. So the example micrographs, some of them are good ones, but most of them are really bad ones. And plotting the histogram of CTF resolution for all micrographs, also, you can also see that many of them are really bad ones. So the higher this number, the worse the quality. So many of them are with, so many of the micrographs here are with bad quality, but there are still some of them that are lower than 5 or 6, which indicates decent quality. So we use a few data sets, basically a few squares for training, and then we use the rest of the data sets for validation or for real. So basically, we apply our trained model on top of it. And we can plot the trajectory the cryo-RL decides to take micrograph from. And it didn't really select a lot of squares. And when we zoom in the trajectory, we can see it only visited 1, 2, 3, 4, 5, 6 different squares. And we can zoom in these squares to see what's really happening inside there. So this is a trajectory overlay on the square level image. And when you zoom in over here, you can clearly see where it's collected the data set from, and the trajectory is panning. And because we also have the ground truth of almost all the holes here in the square, so we can overlay the ground truth on top of this image and also on top of our trajectory. And here we can see cryo-RL decision making, and it successfully avoided most of the bad holes, which are this patch and this patch and this patches. And it'll try to collect as many of the data as possible, but also to reduce the time wasted with moving across different patches. So example here would be this patch. So this patch contains maybe only one good image here, but the algorithm decided not to visit this patch because it'll waste a lot of time to actually collect on this and then move to a different patch, which reduce the reward and also increase the time cost. So also it will make some mistakes, but that's mainly due to the imperfect of the regressor's prediction. So how do we evaluate or how do we compare the results from cryo-RL? We use a very naive baseline to compare this with. So basically, we start from a random position, and then a naive policy will just collect as many as micrograph as possible in a given time limit. So for the naive policy, we don't optimize for the data quality, but we optimize for the number of the micrographs collected. So we also have some quantifications of cryo-RL's performance. This is the trajectory showing the first 100 micrographs collected by cryo-RL. And this is, we try to collect the data set as below 6 angstrom resolution. So as you can see, although it makes some mistakes, but most of the data collected are below 6. And this is the histogram of cryo-RL versus the all micrographs. So the all micrographs as the candidates has many poor or bad or codes with bad qualities. But cryo-RL is enabled to enrich the parts which is below 5 angstrom resolution. And this is also shown here, whereas the baseline is the naive baseline I introduced before, and the cryo-RL's performance is plotted on the right. And this is plotting the number of the micrographs during an 8-hour simulated during an 8-hour time duration for a simulation. And the cryo-RL here shows collected micrograph with much higher quality, overall quality compared to the baseline. So it's it doubles the micrograph numbers for the micrograph that's lower than 4 angstrom, and also it's collected a lot more micrographs with lower than 5 angstroms. And later we also plot the behaviors, so some high-level behaviors analysis of the cryo-RL trajectory. It totally collected over 400 micrographs, and it collected around 75 micrographs per square. It only visited around 50 unique patches, only 6 squares visited, and only 5 unique grid areas visited. And we also compare this with the policy network we trained with different threshold numbers. So if you still remember the reward table, this defines, so this threshold number defines the strictness of the reward table. When, for example, you get a maximum reward when t is equal to 6, you'll get a maximum reward when you collect the data set, when you collect the micrograph with lower than 6 angstrom resolution. But when t equals to 4, you get the maximum reward only if you collect the data, a micrograph with lower than 4 angstrom resolution. So we can see here that a lower t value, which means a more strict reward table, will encourage the policy to collect less micrograph per square, and it'll encourage the policy to explore more across the square. So it'll visit a lot more unique patches, a lot more squares, and a lot more grid areas. And because it wastes a lot of time during the searching, during moving the scope to a different area, so it'll result in collecting lower micrograph numbers totally. So in real practice, so what I've shown before is when the regressor and also the policy, the DQ network is trained on a subset of the data itself. But in the real practice, you don't want to say, I have to systematically collect a few thousand micrographs and train some models and apply on the rest of the data set. So we need to prove the transferability of the models and of this framework. So can we train cryo-RL on a different data set and say on an elderly data set and apply on a different data set? So we collected this apoferritin data set on a different type of grid. This is gold grid. And similar as before, we also collected as systematically, so obtain all the ground truth for all the squares here. We end up with 34 squares and over 3,000 holes. And here we use the deep regressor, which is a general model that I introduced before, training on over 100,000 of images. And you also use the policy network, which I'm trained on the elderly, which is the data set I shown before. So both models are transferred from another data set, and this is the deep regressor performance from the general model. You can see this is like a poorer for performance compared to the ones shown before. but it still captures the overall trend of along this diagonal line. But the good thing about this framework is that you don't need to have a perfect regressor to collect efficiently. So this is the first 100 micrographs collected by cryo-RL, and this is the overall distribution of data quality, of distribution of CTF resolution. And you can see that the ground truth, so the data quality for all micrographs, basically a lot of them are not good ones, but cryo-RL has managed to accumulate most of the good ones in here. Also shown here is that when comparing to the naive baseline, cryo-RL is able to collect more efficiently with much higher much higher number of micrographs compared to the baseline with good qualities. And also we observe similar patterns from the behavior plots where a lower t-value will encourage the policy to explore more areas across the grid and result in collecting less number of micrographs totally. So the last experiment I'm going to talk about is cryo-RL compared directly with human, with an expert, which is, so we collect this data set, we obtain the atlas, and we carefully divide the atlas as fairly as possible into halves. The top part is collected by cryo-RL. So we do systematic data collection and run cryo-RL on top of it. And the lower part is collected by Mike, which is my PI, as an expert for the baseline, for comparison. So for the cryo-RL part, we still use a general regressor and the policy network trained on another data set, and we do data collection. So both models are transferred. And for the lower part, Mike would just freely collect a few over 5,000 or over 500 micrographs on the other half of the grid. So this is the first 100 micrographs collected by cryo-RL and also by an expert. So as you can see, an expert makes multiple mistakes, but so is cryo-RL. But when we plot the number of the micrographs across the total trajectory, and this is when an expert, the expert trajectory translated to our time cost table. So during an 8-hour time duration, we can see that cryo-RL performs much better than the expert and much better than the baseline. And the expert actually failed to identify the squares which contains CTF resolution below 4 angstroms. So it performs even worse than the baseline because the expert did not have the global information when it started the data collection here. Whereas in cryo-RL, it has the global information with all the candidate host predicted CTF values. So it's able to efficiently accumulate the data, the micrograph with high quality. And also plotted here is the behavior radar plot, where the cryo-RL behavior as shown so the behavior of cryo-RL with different t-values exhibit a similar pattern, where a lower t-value will encourage the policy to explore more areas. But more interesting is when you see the expert actually visited a lot more different unique grid areas in the early stage of the data collection, because for an expert, it needs to start from one area, dive down to the microscope level, dive down to the micrograph level, and then determine whether this is a good hole or determine whether this is a good square, and determine basically it needs to search for the parts with better data quality. So the conclusions, reinforcement learning combined with whole regressor allows successful or more efficient data collection compared to an expert. And cryo-RL learns a policy for collecting images that maximize data quality given a limited time. And parameter setup allows for relaxed or more stringent data collection basically you can tune the parameter to encourage the policy to visit more areas if you are doing like a screening session. Some future directions, we plan to do more cryo-RL versus expert comparison to have more data points basically. And another part is active learning to update the regressor during data collection, because as we collecting data, we learn more about the data quality relationship between the square level between the whole level images. And we can use active learning to update the regressor during the data collection to predict more precisely with higher accuracy. And that'll help with the trajectory planning for the policy network as well. And next, another thing is we probably need a better metric for a good micrograph. Currently, we are using CTF resolution, but as introduced before, it also has its own limitations. And also, how do we know when to stop data collection? If, say, we have a very bad grid that's basically no good data on it, and what's the early stop point when that we say that before the given time duration, how do we call, say, this is not a good grid, and I'll just stop collecting. The last thing is to incorporate this into current data collection softwares like cryo-EM or Legion. And we're looking for grad student, postdoc, and collaborators on this project and on other side of the projects in the lab. So acknowledgments, this is a collaborative work with Seychelles Voss from MIT, my PI, and Cianfrocco, especially. He's the main contributor of the algorithm and the experiment design together from IBM. And Cianfrocco lab, especially Nick and Lily for making grids, cryo-EM facility for letting us use the scope for this systematic data collection, and NIH and NSF fundings for this project. And I'd like to thank everyone and happy to take any questions.
Yeah. If anyone has questions online, again, you can put them in the chat box and I can read them out, or you can use the Zoom reacting function, raise your hand, and call on you. While we're waiting to see if anyone has any questions, I actually have a quick question. You mentioned that you plan to do some more testing with expert cryo-EM people. I assume that would involve experts outside of your cryo-RL team.
Yes.
It seems to me like there could be an argument that
Yes.
you're a little biased as an expert.
Yes, exactly.
Yeah.
And also such experiments are expensive to do because collecting a data set like systematically it takes 3 or 4 days at least. So we are planning to take use a simulator to mimic the data collection process. So it saves time and also saves money, and we can ask other experts. We can basically distribute the software to other experts and gather their trajectory from it.
Great. There are no questions yet, but I know sometimes it takes people a little time to type in, so we'll give it a few minutes to keep